The next 6 tests will be predominantly NCERT based. Right? Today also was like that. Why are you laughing? Why everybody is laughing at it? So there is some common joke going on. What happened? What happened? Come on, tell me also. What happened? What? Am I the only one missing something here? And the pain of everybody knowing something and you not knowing anything. Please. What is it? What happened? Come on, you're also laughing, so tell me. Yes, sir.
You have been telling this many times, but still questions are coming from out of syllabus.
Still questions are coming from out, but it has reduced, no?
Only today questions answered it. But previous questions are out of syllabus.
Exactly. Tell me about that in the next test also. Next test you tell again. See, some topics I don't want to leave out. See, let it be. Let it be. Got it? We can't dispute a question if it comes, because some important topics can be related. You can say it is related to something. Okay. Right. Come on. 136. DNA present in chloroplast is, both chloroplast and mitochondria have DNA. Extranuclear DNA are present in 2 organelles, plastids and mitochondria. The nature of DNA is, what is the kind of DNA that is present in chloroplast and mitochondria? Both circular and double-stranded. Circular and double-stranded. Do we find the presence of single-stranded DNA as genetic material? Single-stranded DNA of genetic material in eukaryotes? DNA in eukaryotes is always double-stranded. I mean, in bacteria also, it is double-stranded only. In viruses, you can find single-stranded. Okay. Which of the following are fused in somatic hybridization involving 2 varieties of plants? Protoplast, pollen, callus, somatic embryos. What are fused? Protoplast fusion. Somatic hybridization is brought about by protoplast fusion. Can we combine somatic embryos? If you combine somatic embryos, can we combine somatic embryos in the first place? Somatic hybridization will give rise to a somatic embryo. Correct? Somatic hybridization will give rise to a somatic embryo. Can you combine 2 cells from the somatic embryos and still make another viable embryo? Is it possible? If you do that, what would that be? If you do that, what would that be? The wheat that we consume now is, what is the ploidy of the wheat that is commonly available in the market now? Hexaploid wheat. We have hexaploid wheat now. Polyploidy is achievable in plants. Polyploidy is a way of getting newer varieties in plants, newer species, newer varieties for the same species. But polyploidy is not viable in animals. Polyploidy is not viable in animals, it is viable in plants. Callus gives rise to, callus is the way of getting what? Anything that the plants that you get from the differentiation of callus would be clones of the parent plant. They are clones. They are exactly similar in genetic composition. Exactly similar in genetic composition. Genetic clones. What is the chemical that you use for bringing about protoplast fusion? When you are talking about protoplast fusion, you should also recall the chemical that you will use. What is the chemical that you will use for protoplast fusion? PEG. What is PEG? Polyethylene glycol. Polyethylene glycol is PEG. Somatic hybridization is brought together. You have to make note of those things. Whenever I am telling that, you have to make note of it. If you don't make note, how do you revise? You should put it into your notes. That is the revision tool, revision material that you build. Identify the correct description about the given figure of a plant inflorescence. What inflorescence is that? Clistogamous flower showing autogamy. Is it clistogamy that is shown there? It's not a clistogamous flower. What is a clistogamous flower? Clistogamous flowers are closed flowers that do not open. It is not clistogamous flower, it is open. So many anthers are visible. Compact inflorescence showing complete autogamy. It is not autogamous at all. Wind pollinated plant inflorescence showing flowers with well exposed stamens. Water pollinated flowers showing stamens with mucilaginous covering. It looks like a wind pollinated plant. What is the technical term for pollination by wind? Anemophily. Anemophily. Repeated reference to these terms and concepts are necessary. Keep repeating till the time it becomes so ridiculously common. So what is special about anemophilous flowers? Okay, let us say anemophilous flowers and entomophilous flowers, entomophilous plants. Which one will have to put in more energy for reproduction? Which one will spend more energy for reproduction, for pollination? Entomophilous. Why? They have the additional pressure of creating colorful, fragrant and nectar-filled flowers. Anemophilous flowers need not do any of those things. They just have to produce more stamen. Even the entomophilous plants have to produce more stamen. There is no choice there. They have to produce stamens in abundance. Spraying sugarcane crop with which of the following plant growth regulators increases the length of the stem, thus increasing yield? Don't look at the options. Which is the chemical which can increase the length of the stem? Stem elongating hormone in plants. Gibberellin is stem elongating hormone. The disease associated with that, which resulted in the discovery of gibberellin. What is the disease? Foolish seedling or foolish sapling disease. What is the technical term given for the Japanese term for the foolish sapling disease? Bakane. Alright, foolish sapling disease. The foolish saplings which grow under the influence hyperstimulation of gibberellin produces more yield. True or false? They produce more yield. True or false? They grow fast, wilt and die. They don't produce any yield. So gibberellin. If you have to give one word to cytokinin, what would you give? One major function for cytokinin. Like gibberellin is stem elongating hormone. Auxin is apex elongating hormone, apical dominant. Cytokinin promotes cell division and differentiation. Then abscisic acid, hormone of breakdown, abscission, growth inhibiting hormone. What is the fungus? Fungus which produced gibberellin. Fungus that produced gibberellin. Gibberella fujikuroi. Okay, scientists and names. Which one do you want to catch hold of? Griffith, Jacob and Monod. Jacob and Monod is black operon concept. So B4, only one. Option 4, A3, Frederick Griffith transformation. Griffith's transformation experiment. In which organism did he conduct the transformation experiment? Griffith. Which organism? Which is the organism in which transformation experiment was conducted by Griffith? That can also be a question, that is a more relevant question than the name of Griffith. Mice. Mice was infected with this organism. Streptococcus pneumoniae. R strain is virulent or S strain is virulent? In Streptococcus pneumoniae, S strain. The smooth bacteria are virulent. So transformed bacteria would be smooth or rough. Transformed bacteria would be smooth or rough in the context of Griffith's experiment. Smooth. Okay. Again, matching glucose transporter 4, insulin, trypsin, collagen. Okay. Glucose transporter 4, what does it do? It enables glucose transport into cells. A4, insulin, hormone, trypsin, enzyme, collagen, intercellular nerve substance. Is very easy question. What is special about trypsin as an enzyme? You should be able to tell this by virtue of attempting so many out of syllabus questions. What is special about trypsin as an enzyme? Trypsin is a special enzyme. Trypsin is autocatalytic. Trypsin is an autocatalytic enzyme. It can catalyze the conversion of trypsinogen into trypsin. Enterokinase is the enzyme which is produced by duodenum, which acts on trypsinogen to produce the initial stock of trypsin. And once trypsin is produced, it can do autocatalysis. It can convert the inactive trypsinogen into trypsin by itself. Statements, correct statements we have to identify. Statement 1 and 2. In C3 plants, some oxygen binds rubisco, hence carbon dioxide fixation is decreased. Is it true? It is true. What is that process called? Where rubisco binds to oxygen and breaks down the fixed carbon compounds. What is it called? Photorespiration. Photorespiration reduces the yield of carbon fixation. Photosynthetic products are reduced because the same products are used by rubisco to break it down by utilization of oxygen. So statement 1 is correct, true. Statement 2, in C4 plants, mesophyll cells show very little photorespiration, while bundle sheath cells do not show photorespiration. True or false? False. Why? In mesophyll cells in C4 plants, there is no risk of photorespiration because in mesophyll cells there is fixing of carbon into what? Carbon is fixed into? What is it fixed into? Oxaloacetic acid, and then malate stored. Then malate is transported, malate and oxaloacetate is transferred into the bundle sheath cells where rubisco is present. So rubisco is not getting exposed to the oxygen that is produced. Why photorespiration happens? In the same mesophyll cells, light reaction is also happening, and light independent reaction is also happening. Carbon fixation is also happening. Rubisco is a dual enzyme. It can bind carbon dioxide and fix carbon dioxide, or if oxygen binds to it, it can break carbon products into carbon dioxide. And that requires oxygen to be present in that cell. When light reaction and dark reaction, both of them are happening in the same cell, there is possibility of photorespiration. But in C4 plants, light reaction happens in the mesophyll cells, and dark reaction happens in the bundle sheath cells. What is the enzyme? What is the enzyme you have to remember in C4 plants, in the mesophyll cells? PEP carboxylase, phosphoenol pyruvate carboxylase. PEP carboxylase you have to remember for C4 plants. Identify the step in the TCA cycle which does not involve oxidation of substrate. When we say oxidation of substrate, what does that mean? Some energy compound is produced, something is removed. Alright? Succinyl CoA to succinic acid. Is there any energy reaction happening there? Succinyl CoA to succinic acid, anything is being produced? GDP is produced, but is there any breakdown of succinyl CoA? Any oxidation happening there? No. Isocitrate to alpha ketoglutarate, and what is the number of carbon atoms in succinyl CoA and succinic acid? 4 carbon. Isocitrate to alpha ketoglutarate. Isocitrate, how many carbons? 6 carbons. Alpha ketoglutarate, 5 carbons. Malic acid to oxaloacetic acid. Both are 4 carbons. Both are 4 carbons. NADPH is produced in that reaction also. Isocitrate to alpha ketoglutarate also, there is NADPH produced. Succinic acid to malic acid. Succinic acid to malic acid, again 4. FADH2 is produced there. So what is the ATP synthesis in the step of succinyl CoA to succinic acid called? What phosphorylation is that? What phosphorylation is happening in the conversion of succinyl CoA to succinic acid? Substrate level phosphorylation. Phosphate group is added. Phosphate group is added to adenosine, not adenosine, let's say, during the formation of nucleotides. During the formation of nucleotides, you take a base, you take a phosphate, and you take a sugar. And sugar, phosphate, and base. Phosphate group is added to the sugar. Do we call that also as phosphorylation? Is that also phosphorylation? Is that also phosphorylation? No? Should we consider that also as phosphorylation? Deoxyadenosine is produced. Deoxyadenosine. Is it phosphorylation there? One phosphate group being added? You can still consider that phosphorylation, that is substrate level phosphorylation. In the reaction, in any reaction where a substrate is getting converted into product, you attach a phosphate to that substrate level phosphorylation. Not necessarily an energy substrate is being produced. ATP is what? Adenosine triphosphate. What is adenosine? Adenosine is a nucleotide only. Matching again, citric acid cycle, glycolysis, electron transport system, proton gradient, where each one is happening. Citric acid cycle happens in. Don't look at the options. Now the question is, list 1 is the process, are the processes, list 2 is the site where anything happens. So without looking at it, tell me where citric acid cycle happens. In the mitochondria. Where in the mitochondria? Mitochondrial matrix. Glycolysis happens in the cytoplasm. Electron transport system is present in the inner mitochondrial membrane. Proton gradient is found in the, is created in the intermembrane space. Intermembrane space. Where is the proton gradient created in chloroplast? Where is the proton concentration happening in chloroplast? Inside the where? The gradient is inside to outside or outside to inside? Proton is concentrating in the stroma of the chloroplast and it moves outwards. Through what? What the proton moves out through? Proton during oxidative phosphorylation or photophosphorylation moves through a protein complex referred to as, what do you refer to it? Refer to it as ATP synthase. ATP synthase is the protein complex through which the protons move, and the change in the configuration of the ATP synthase will bring substrate and phosphate together. Binding of that will lead to ATP synthesis. So, again, whenever we look at electron transport chain, four things you have to remember. Who is the electron donor? Electron transport chain in mitochondria. Who is the electron donor? Who is the electron donor and who are they donating it to? Come on, you cannot forget these things. Who is the electron donor for electron transport chain in mitochondria? NADPH and FADH2. NADPH donates electrons to which complex? Complex 1. FADH2 donates electrons to complex 2. Which of the complexes among complex 1, 2, 3, 4 does not contribute to creating a proton gradient? Which complex among the 4 does not contribute to creating a proton gradient? Complex 2. This is the membrane. Complex 1, complex 2, complex 3, complex 4, and you have the ATP synthase complex. ATP synthase complex. 1, 2, 3, 4 and ATP synthase. What do you have in between here? Mobile complexes. What do you have here? What do you have here? Huh? Plastoquinone is in. This is ubiquinone. Here, mobile complex is ubiquinone. Here, another mobile complex is cytochrome C. That's all. When it comes to C, the entire explanation of how it moves and all is fine. But what you have to remember, NADPH donates electrons in complex 1. FADH2 donates electrons to complex 2. Ubiquinone is the mobile carrier from 1 to 3 and 2 to 3. ATP synthase. That's all. So whenever electrons flow through a transmembrane complex, it will lead to the expulsion of a proton. So which ones are the transmembrane complexes? 1, 3, 4. These 3 are transmembrane complexes. So when electrons flow through one, proton expulsion, proton expulsion, proton expulsion. Gradient is created on the outside of the inner membrane. This entire thing is the inner membrane. And proton comes back through this, producing ATP. Electrons are flowing, and when they come to the end of it, somebody should receive the electron. That is the terminal electron acceptor. So, why are electrons flowing? Electrons should flow because you need to create a proton gradient. You have to pull hydrogen ion from the matrix and push it outside of the inner membrane. You need energy for that. That energy is provided by the flow of electrons. when electrons are flowing, they are moving from a high energy state to low energy state and finally they died out. Somebody is receiving it. Correct? When the electrons are moving from a high energy state to low energy state, energy is released. That energy is used for pushing out protons. Is it understood? And that proton comes back to produce ATP by using that energy. When it flows through, ATP is produced. So 4 things you have to remember. Electron donors are NADPH and FADH2. NADPH donates to complex 1, FADH2 donates to complex 2. Second, proton gradient is created outside of the inner membrane. That is in the intermembrane space. Here you will have the outer membrane. So this is the intermembrane space. Oxygen is the terminal electron acceptor. Oxygen is the terminal electron acceptor. Clear? That's all. Electron should flow. Who is the donor? Who is the acceptor? What is the direction of the gradient? That much is enough. If you remember that much, you should be able to answer good number of questions that come based on electron transport chain. The questions will not come asking you what all are there inside this. Asking you for the series of complexes that will carry. Alright, cytochrome, sometimes you will get questions asking the sequence of cytochromes, BCA, Bachelor of Computer Applications. Cytochrome B, cytochrome C and cytochrome A. That's all, BCA. Similarly, in chloroplast, during photosynthesis, who is the electron donor? Who is the electron donor? There are 2 photosystems there. It is a Z complex, Z structure. Okay. This is the, you have PS1, then you have, here you have your item. What is that? Plastoquinone. Then you have photosystem 1, this is photosystem 2. Then you have FNR, NAD reductase. That's all. Alright. So here you have photolysing complex. Photolysing complex is the hydrolysis complex. Electrons are triggered from photosystem 2, goes to plastoquinone. Through a series of things, it will go to photosystem 1, then it goes to FNR. Ferredoxin, NADP reductase. Ferredoxin, NADP reductase. See, this is the Z scheme or the Z scheme. So, who is the electron donor for photosystem 2? Water. Breakdown of water will give you electrons to top of the electrons. When electron is lost, somebody has to give it, no? Otherwise, how can you keep removing electrons from an orbit? You cannot keep removing electrons from something unless you give the lost electron. Give the lost electron. You always, the electron balance should be maintained. Why is that? Removing electron from its orbit requires what? Energy. Electrons are held, they are pulled towards the nucleus by the nuclear force. You have to supply energy externally for the electrons to attain sufficient energy to just get out of the influence of the nucleus of that particular atom. So once that is removed, you remove one electron. If you have to remove another electron from the same atom, can the same amount of energy do that? Can the same amount of energy remove one more electron? Not possible, because that is just 3-4 points. Photophosphorylation, oxidative phosphorylation. Photophosphorylation, oxidative phosphorylation. Electron donor. Who is the electron donor in photophosphorylation? Water. In oxidative phosphorylation, NADPH and FADH2. Alright. Electron acceptor. In photophosphorylation it is NADP. Here it is oxygen. H plus gradient. Inside to outside. Oh. So I was thinking there is something wrong here. In oxidative phosphorylation, gradient is outside to inside. That's all. This small table you should remember. See, I understand it is not easy. It is not easy to remember everything. You have to back yourself up to arrive at answers when you attempt the questions. Okay. If you go with an approach that I should remember everything before I go into the examination, difficult. Go into the examination, try to come to the answer. Your intuition will help, your practice will help. Is this clear? This much. Rest all, what is the sequence of compounds, what is the sequence of electron carriers, all of that, you can still not remember. It's okay. Which of the following statements is correct regarding the process of replication in E. coli? Replication in E. coli, correct statement we have to find out. DNA-dependent DNA polymerase catalyzes polymerization in 5 to 3 as well as 3 to 5 direction. Replication in E. coli. So we are talking about DNA replication basically because all the 4 options are related to DNA. DNA-dependent DNA polymerase catalyzes the polymerization in 5 to 3 as well as 3 to 5. Is it correct? Whether it is bacteria or any of the eukaryotes, unicellular or multicellular, the direction of replication or anything is 5 to 3. 5 to 3 is the direction of movement. So, 3 to 5 direction is wrong. But is the first half correct? Is it DNA-dependent DNA polymerase that acts on it, that causes polymerization? That part is correct. Second statement, DNA-dependent DNA polymerase catalyzes polymerization in 5 to 3 direction. That is correct. Again, 3 to 5 is the third option, wrong. DNA-dependent RNA polymerase is given in the fourth option. That part is wrong, while the second part is correct. So, statement 2 is the correct statement. DNA-dependent RNA polymerase is it involved in anything? DNA-dependent RNA polymerase, is it significant? Is it associated with anything in the bacteria? What? Where do you need DNA-dependent RNA polymerase? For what do you need it? Transcription. You need to make an mRNA, no? You need an RNA polymerase which will read the DNA and create a complementary copy to that. Direction of the template strand. Just 3-4 points. Photophosphorylation, oxidative phosphorylation. Photophosphorylation, oxidative phosphorylation. Electron donor. Whose electron donor is photophosphorylation? Water. In oxidative phosphorylation, NADPH and FADH2. Alright. electron acceptor. In photophosphorylation it is NADP. Here it is oxygen. H plus gradient. Inside to outside. Oh. So I was thinking there is something wrong here. In oxidative phosphorylation, gradient is outside to inside. That's all. This small table you should remember. See, I understand it is not easy. It is not easy to remember everything. You have to back yourself up to arrive at answers when you attempt the questions. Okay? If you go with an approach that I should remember everything before I go into the examination, difficult. Go into the examination, try to come to the answer. Your intuition will help, your practice will help. Is this clear? This much. Rest all, what is the sequence of compounds, what is the sequence of electron carriers, all of that you can still not remember. It's okay. Which of the following statements is correct regarding the process of replication in E. coli? Replication in E. coli, correct statement we have to find out. DNA dependent DNA polymerase catalyzes polymerization in 5 to 3 as well as 3 to 5 direction. Replication in E. coli. So we are talking about DNA replication basically because all the 4 options are related to DNA. DNA dependent DNA polymerase catalyzes the polymerization in 5 to 3 as well as 3 to 5. Is it correct? Whether it is bacteria or any of the eukaryotes, unicellular or multicellular, the direction of replication or anything is 5 to 3. 5 to 3 is the direction of movement. So, 3 to 5 direction is wrong. But it is the first half correct. Is it DNA dependent DNA polymerase that acts on it, that causes polymerization? That part is correct. Second statement, DNA dependent DNA polymerase catalyzes polymerization in 5 to 3 direction. That is correct. Again, 3 to 5 is the third option, wrong. DNA dependent RNA polymerase is given in the fourth option. That part is wrong when the second part is correct. So statement 2 is the correct statement. DNA dependent RNA polymerase is it involved in anything? DNA dependent RNA polymerase, is it significant? Is it associated with anything in the bacteria? What? Where do you need DNA dependent RNA polymerase? For what do you need it? Transcription. You need to make an mRNA, no? You need an RNA polymerase which will read the DNA and create a complementary copy to that. Direction of the template strand during transcription. Polarity of the template strand during transcription. 3 to 5 or 5 to 3. Template. Template is 3 to 5 because the mRNA should be built from 5 to 3. Anti-sense strand direction. Anti-sense strand direction. 3 to 5 again. Sense strand direction is 5 to 3. mRNA is an exact copy of sense strand or anti-sense strand. Sense strand. mRNA is a complementary copy of anti-sense strand. So question can be anything, sense, anti-sense, It can include both sense strand, anti-sense strand, template strand, coding strand, mix and match, give some 2-3 combinations. If you just know mRNA should be an exact copy of the gene and the direction is 5 to 3, you can arrive at the answer. You should think. And here when we talk about DNA, lagging strand, leading strand, direction of the template for leading strand. During DNA replication, what is the direction of the template complementary to which the leading strand is built? 3 to 5, good. Direction of the template against which Okazaki fragments are built? 5 to 3. That's all. The approach is to fix in your mind that it is 5 to 3, 5 to 3, 5 to 3, that's all. So whatever is the question, build a response to that in your mind. The right approach is look at the question and arrive at the answer. Don't look at the question and try to recall the answer. Okay? In an ecosystem, if the net primary productivity of the first trophic level is 100x, what would be the gross primary productivity of the third trophic level of the same ecosystem? First trophic level is 100. So second trophic level would be? Second trophic level would be what? What law is being tested here? Your knowledge of which law is being tested here? The bioenergetic law or the 10% law. Correct? Who is the scientist? Lindemann. Lindemann's law, na? 10% law. So what does that mean? The amount of energy being transferred from one trophic level to another trophic level is 10% of what is available in the previous trophic level. So if 100 units of energy is available in the first trophic level, what will be the amount of energy that will be fixed in the second trophic level? 10% of 100 is 10. What would be the energy fixed in the next trophic level? 1. So if 100x is the net primary productivity of the first trophic level, what is the gross primary productivity of the third trophic level? Third trophic level would be x. This is the x kilocalories per square meter per year. So here gross primary productivity, net primary productivity, secondary productivity, all of this is important. Net primary productivity considers loss of energy in the form of dash. Net primary productivity is obtained by removing the loss of energy from gross primary productivity in the form of respiration. Respiratory losses is removed. What about defecation? An animal is consuming something. Let's say a deer is consuming a plant. Some of it is absorbed and some of it is lost in the form of defecation. Now how do we consider the amount of energy fixed by this? The entire energy that was available in the food that the deer ate did not get converted, did not get absorbed, no? Is it including that? If 100 grams of plant is consumed by the deer, or 100 units of energy in the form of mass is consumed by the deer, some of it is lost in the form of defecation also. Including that, is it 10 units getting fixed in the body? So do we have to consider the defecation loss? Considering all that, we are not talking about how that 90% is going away. We are talking about the transfer of biomass or the transfer of energy in the form of fixation of mass from one trophic level to another trophic level is 10%. 10% gets transferred as mass into the next trophic level. How did the rest of the 90% go? That takes into consideration the defecation loss also. Matching rose pea, cotton, mango, estivation. What estivation is rose? That by common sense logic we can say. Twisted estivation, one in, one out. The rose petals are arranged like that. So rose is twisted estivation. So it's given in the same order, is it? Rose is twisted estivation only, no? So 1, 2, 3, 4. P perigynous flower. What is perigynous flower means? What is a perigynous flower? A flower in which the ovary is in line with the other whorls. The level at which the ovary is present is the same as that of the other whorls. Next question, May, Humboldt, Paul Ehrlich, David Tillman. All people associated with ecology. Which one do you want to catch hold of? Alexander Humboldt, what did he give? Global species diversity is about 7 million. Is that what Humboldt gave? Yeah. So B is 3, B3. What? B1. Okay. Yeah. Humboldt, species area relationship, then filariasis is caused by a nematode. Filariasis is caused by a nematode. What is the parasite there? Filariasis. 2 parasites you have to remember. Vector is what? Which mosquito? Culex mosquito. Culex mosquito is the vector for filariasis. 2 pathogens you have to remember. One is Wuchereria bancrofti and the other one is Brugui malai. Wuchereria bancrofti and Brugui malai. 2 vectors, 2 pathogens. Again matching the questions. Non-medicated IUD, which of the 4 is non-medicated? Lipis loop is non-medicated. Copper releasing IUD. Copper releasing IUD is multi-load 375. Hormone releasing IUD is hormone releasing IUD. Progestogens is it an IUD? Progestogen, is it an IUD? Hormone releasing IUD. Progestogen. Anyways, we have to consider that only. Implant is of LNG20, levonorgestrel. Implant is levonorgestrel. LNG20 is the hormone releasing IUD, isn't it? LNG20 is the hormone releasing IUD. Levonorgestrel 20 is the hormone releasing IUD. Implant is of progestogens. Progesterone implant. Low doses of progesterone being released. So which option matches that? Non-medicated IUD, lipis loop A3. Okay. B1. Oh, it matches. C4, D2. Option 2. Statements, correct statements. Presence or absence of the hymen is not a reliable indicator of virginity. True or false? What is hymen? Hymen is a fibrous covering at the opening of vagina. It's a thin fibrous covering at the opening of vagina. If it is present, it will be ruptured during the first sexual intercourse. But it is not only sexual intercourse which will lead to the breakdown of hymen. Physical activity, intense physical activity, running, cycling, swimming, exercising, all of these can lead to rupture of hymen. You know, there was this pathetic practice of checking women for virginity before allowing them to enter Europe. You know this? Especially women from South Asia, India, Pakistan, and these countries. Women were encouraged to enter Europe to get married to Europeans and become Europeans. What nonsensical practice was that? It was abandoned. It was banned in 1979. So till 1979, this was a practice, checking women for virginity. And who would do it? The immigration officials at the airport. So women who are traveling from India to Europe would be stripped and they would be checked for the presence or absence of hymen by immigration officials. And these are the people who will tell us that they will civilize us. Very rightly, they are going down. Karma comes back. You go around the world destroying everybody and looting everybody. Now you are going down and down and down, and not very long before you become a beggar. So I came to know about this historical fact about a few days ago. I was shocked. Filariasis is caused by a nematode. Filariasis is caused by a nematode. What is the parasite there? Filariasis. Two parasites you have to remember. Vector is what? Which mosquito? Culex mosquito. Culex mosquito is the vector for filariasis. Two pathogens you have to remember. One is Wuchereria bancrofti and the other one is Brugui malai. Wuchereria bancrofti and Brugui malai. Two vectors, two pathogens. Again matching the questions. Non-medicated IUD, which of the four is non-medicated? Lipis loop is non-medicated. Copper releasing IUD. Copper releasing IUD is multi-load 375. Hormone releasing IUD is hormone releasing IUD. Progestogens is it an IUD? Progestogen, is it an IUD? Hormone releasing IUD. Progestogen. Anyways, we have to consider that only. Implant is of LNG20, levonorgestrel. Implant is levonorgestrel. LNG20 is the hormone releasing IUD, isn't it? LNG20 is the hormone releasing IUD. Levonorgestrel 20 is the hormone releasing IUD. Implant is of progestogens. Progesterone implant. Low doses of progesterone being released. So which option matches that? Non-medicated IUD, lipis loop A3. Okay. B1. Oh, it matches. C4, B2. Option 2. Statements, correct statements. Presence or absence of the hymen is not a reliable indicator of virginity. True or false? What is hymen? Hymen is a fibrous covering at the opening of vagina. It's a thin fibrous covering at the opening of vagina. If it is present, it will be ruptured during the first sexual intercourse. But it is not only sexual intercourse which will lead to the breakdown of hymen. Physical activity, intense physical activity, running, cycling, swimming, exercising, all of these can lead to rupture of hymen. You know, there was this pathetic practice of checking women for virginity before allowing them to enter Europe. Do you know this? Especially women from South Asia, India, Pakistan, and these countries. Women were encouraged to enter Europe to get married to Europeans and become Europeans. What nonsensical practice was that? It was abandoned. It was banned in 1979. So till 1979, this was a practice. Checking women for virginity, and who would do it? The immigration officials at the airport. So women who are traveling from India to Europe would be stripped and they would be checked for the presence or absence of hymen by immigration officials. And these are the people who will tell us that they will civilize us. Very rightly, they are going down. Karma comes back. You go around the world destroying everybody and looting everybody. Now you are going down and down and down, and not very long before you become a beggar. So I came to know about this historical fact about a few days ago. I was shocked. The justification given by Great Britain for colonization was civilizing people from the third world countries. And why did the countries become third world countries? Because these people colonized and looted them. In both sexes of cockroach, a pair of jointed filamentous structure called cerci are present on which segment? 10th segment. What is present only in males in the 10th segment? Anal style. Brain structure and functions. Don't look at it. Go from the brain stem to the higher centers. If we go from the lower part of the brain stem to the higher centers, what is the first one you come across? The lower part of the brain stem is medulla. What is it useful? What is it known? What is it for? Vital centers, respiratory center, and vasomotor center. Then you have midbrain. Midbrain have important nuclei where the consolidation of fibers happen. Pons is called the bridge between the brainstem and the higher centers. Then you reach the thalamus. Thalamus is the relay center. The fibers are relayed to the higher centers, to the relevant centers by bundling it in the respective bundles. Then after you move from thalamus, you have go further higher. You have all the limbic system which are associated with memory, emotions, and all that. Then you have the cerebrum. Cerebrum has functional areas and association areas. Okay? Functional areas are those which are associated with specific functions. Like you have the speech area, Wernicke speech area and Broca's area. You have the visual cortex, visual areas, auditory areas. Those are all specific for individual functions. That is about 30 to 40% of the brain, of the cerebrum. Bulk of the cerebrum is involved is association areas. What are association areas? They are like processing units, collect inputs from multiple centers, put it all together, understand it in a complex way, and give a response. All of that is brought about by the association areas. Then you have the two balance controlling parts. What are they? They are cerebellum. Cerebellum is for balance. Then you have hypothalamus. Hypothalamus is for chemical control. It regulates you, he told. I'll pull you to court. You do whatever you want. I'm not coming. Then I had shot only for 2 days. They small footages. They used it for 10 episodes to increase my pain. Some of my relatives had watched it. Horrible things. Okay. I could not show my face. Okay. And in that one instance, a kid, my son in that serial, throws something, it hits my head, and I have to just catch hold of it as if it hurt me. This was the scene. You know the scene. It's all explained in the beginning. He threw it, and apparently it had fallen here on the right side. I was catching hold of the left side. Oh. And the director was so angry about my performance. He has not even seen it. Bloody let this fellow go do whatever he wants. And my aunt told me that. She watched it and she told, it's okay, you managed, but the ball hit you on the right side of the road. Why would you catch it? Oh, detailing. It took me a long time to get out of the camera fear. Even now, it takes me some time to shoot in front of the camera when I'm doing technical videos also. The flippers of penguins and dolphins are an example of what evolution? Convergent evolution, divergent evolution, adaptive radiation, natural selection. Convergent evolution, they are not structurally similar, but functionally they are similar. They perform the similar functions. Convergent evolution will lead to the development of what organs? Same, whenever these questions come, you have to recall that. Convergent evolution leads to the development of analogous organs. Divergent evolution will lead to the development of homologous organs. Convergent and divergent evolution will talk with respect to unrelated species. We talk with respect to unrelated species. Developing different kind of adaptations by the same species in response to different environmental stimuli is called adaptive radiation. Is adaptive radiation convergent evolution or divergent evolution? Huh? If you have to brand adaptive radiation in convergent or divergent evolution, which one do you call it? Divergent evolution. But we are talking about divergent evolution in terms of different species, homologous organs. So there is that distinction there, you have to be very clear about it. If at all a question comes where you have to choose convergent or divergent evolution with respect to adaptive radiation, it has to be divergent evolution. But it's not technically the same. And with natural selection, you have to remember, we discussed in the morning yesterday, no? With natural selection, you have to remember radiation. What radiation? What kind of selection? Different types of selection. Natural selection will have stabilizing selection. What is this? Movement? Directional selection or disruptive selection. Remember the graph. Stabilizing selection will lead to smaller extremities. Directional selection will lead to increase in the size of one of the extremities. Disruption will lead to elimination of the mean. The middle average ones will be eliminated. Like if we have to talk about it in terms of human height, humans in terms of height. Most of the people will be between 5 to 5.5 feet. Indian men. Most of the men are between 5 to 5.5 feet height. Or beyond that is lesser number. Below that is also lesser number. If it is directional selection, if more people beyond 5.5 feet are present, then it is directional selection. Something has happened which led to people becoming more tall. Maybe entire generation started consuming Horlicks. Horlicks generation might lead to movement towards one direction. So Horlicks is the evolutionary factor there. If they become shorter, if the entire population becomes on an average more shorter, that is also directional. And if you have only less than 5 feet and more than 6 feet, nobody between 5 to 6 feet, then that is disruptive. Or very few people between 5 to 6 feet, that is disruptive. Just whenever you are looking at questions, just recall all related concepts. Don't just look at the question, look at the options and say what is the right option. No, just try to get something or the other related to that. That exercise you have to do more and more in the next 50 days. Just go here and there. Come back to the question, relate it, go somewhere, come back, go somewhere, come back. Okay. So your revision can be based on your question papers only. You can just take your question paper, look at the questions, go here and there, recall something, have discussions, ask each other. The next 50 days can have more discussion based learning also. Asking questions. You should ask something which you know an answer for. Don't ask something to check what your friend doesn't know. Because when your friend doesn't know that, you should be able to tell the answer. That will be useful. Okay? So don't get that pleasure in making people know that they don't know. There is this joke. They say, let's consider Einstein. Einstein and an Indian were going on a train together. The Indian doesn't know who Einstein is. Einstein is hurt, his ego is hurt. He asks him, do you know who I am? This fellow says, I don't know, I don't care. He says, I am Einstein, I am the most intelligent person on earth. You can ask me any question, I will answer. You can ask me any question, I will answer. But I will also ask you a question, this fellow says, okay, I will also answer your question. They put a bet. If Einstein asks a question and the Indian doesn't know the answer, he has to give him some money. And Einstein is more intelligent, no? He is more egoistic. He says, if you ask me a question and I can't answer it, I'll give you double that money. So Einstein asks a question. This fellow asks a question. He asks a question, what goes on the mountain with 2 legs and comes back with 3 legs? Einstein is, what goes on the mountain with 2 legs and comes back with 3 legs? He thinks and thinks and thinks. He doesn't know. He says, I don't know. He gives some X amount of money. Einstein asks, what is that? This fellow gives half of that money. So who is intelligent here? Einstein is intelligent. We are street smart. I also don't know. Take 5 rupees. He gave me 10 rupees, I gave you 5 rupees. Net profit, 5 rupees. That's all. Okay. So when you are doing discussions, please ask your friend a question for which you know the answer. So when your friend says, I don't know, you tell, and your friend benefits from that. Same. You can make a group of 2-3 people. For the next 50 days, I can allow those groups, small groups, to have discussions and preparation. You can move out of this place also if required. You can sit in small groups and you can do that. Okay. See, now I am positive that you will not be wasting time because we have 50 days left. Next question, lung capacities. Again, you have to tell me what capacity is that I am writing, and you have to tell me the volume also. Baseline. What is this? What is A and B? Both are tidal volume. The volume of air that you inhale after normal exhalation or that you exhale after normal inhalation is this much. So exhale after normal inhalation or inhale after normal exhalation. Both are tidal volume. You can consider it this way. This or this. What is C? And this entire thing is D. What is C? What is C? Inspiratory reserve volume. What is D? What is D? What is D? D is inspiratory capacity. Same on the opposite side. Now this is E is expiratory reserve volume. F is expiratory capacity. Expiratory reserve volume and expiratory capacity. Now this C plus B plus E, from here to here, from here to here, what is it? This is G. G is vital capacity. Vital capacity. Vital capacity, that's all. This you consider as normal inhalation to exhalation. Normal inhalation to normal exhalation. The volume of air that you exhale normally after a regular inhalation is tidal volume. Same on the other side also. The volume of air that you inhale normally after a regular exhalation is tidal volume. Normal inhalation. From there you move to the maximum that you can inhale. That is your inspiratory capacity, inspiratory reserve. After a normal inhalation, what is the extra that you can inhale? That is your reserve volume, inspiratory reserve volume. After you exhale normally, what is the maximum that you can exhale beyond that? That is your expiratory reserve. Reserve is always more than what is available normally. That is reserve. Vital capacity is inspiratory capacity is what? After a normal exhalation, what is the maximum that you can inhale? In that, normal inhalation is also there, no? There is normal inhalation plus whatever you can do beyond that. So inspiratory capacity is tidal volume plus inspiratory reserve volume. Expiratory capacity is tidal volume plus expiratory reserve volume. You understand? So if I write it down as one line, in one line if I write it. Without this graph, this is total vital capacity. You have tidal volume, inspiratory reserve volume, expiratory reserve volume. This whole thing is vital capacity. You see this? Let us say this is A, B, C, ABC. This is ABC. Tidal volume, inspiratory reserve volume, expiratory reserve volume. Inspiratory capacity is A plus B. Expiratory capacity is A plus C. Vital capacity is A plus B plus C. Alright. That's all. If you can just remember this. Tidal volume is A, inspiratory reserve volume is B. So this is our normal respiration. This is what we breathe. What you are doing now is this breathing. So tidal volume is about 500 ml. This is about 3000 ml. This is about 1200 ml. Is that clear? And beyond this, you cannot touch it. That is your residual volume. So you can consider it to be 1250 ml. Symbolically mentioning that residual volume is higher than your expiratory reserve volume. So vital capacity is A plus B plus C, around 6 liters. 5 to 6 liters is your vital capacity. Okay. Is it clear? That's all. This one line will give you all volumes. All volumes. So if I want you to represent vital capacity in terms of inspiratory capacity and expiratory capacity, give me a formula for that. Vital capacity is equal to, I want inspiratory capacity and expiratory capacity also on the right hand side, both on the right hand side. So how do you represent it? Vital capacity is equal to inspiratory capacity plus expiratory capacity minus tidal volume. Why? See, vital capacity is this entire thing. From here to here is vital capacity. Inspiratory capacity is B plus A plus B. Expiratory capacity is A plus C. So A plus B plus A plus C is 2A plus B plus C. What is vital capacity? It is A plus B plus C. So when you mention inspiratory capacity plus expiratory capacity, you have one tidal volume extra there. You have to subtract one tidal volume. That's all. Simple calculations, logical. From here to here is A plus B is inspiratory capacity. Vital capacity is equal to, let us say, inspiratory capacity plus expiratory capacity. Whatever is, this is A plus B plus A plus C. But vital capacity is A plus B plus C. In this, vital capacity is A plus B plus C. But when we have mentioned it as inspiratory capacity and expiratory capacity, it is A plus B plus A plus C. We have one A extra. So what should we do? Minus A. A is what? Tidal volume. Remember this. You remember it like this. Whatever be the question, use common sense and arrive at the answer. We cannot put all possibilities and try remembering everything. This is all you have to remember for volumes. If you remember this, whatever is the question, use common sense. Next. See, whenever I give these kind of things, you should note, you should write down. Alright. And you can help me by sharing those things with me also. Sometimes different, whatever I have not written down myself will be converted. It's all situational. Which one of the following factors will not affect the Hardy-Weinberg equilibrium? Hardy-Weinberg equilibrium is dependent on a few factors. What are the factors? You tell me. When will there be Hardy-Weinberg equilibrium? When the population is large, when there is no migration, when there is no mutation, and when there is random mating. Now look at it. Which of the following factors will not affect the Hardy-Weinberg equilibrium? Gene migration, will it affect? It will affect. There should not be any migration. Constant gene pool, genetic recombination, genetic drift. What will not affect the equilibrium? That means which of the four will not disturb the equilibrium? Constant gene pool. That is a condition for Hardy-Weinberg equilibrium. Again, questions related to Hardy-Weinberg equilibrium will be based on gene frequency, allelic frequency, and phenotypic frequency. Phenotypic frequency. Phenotypic, genotypic frequency. A plus B is equal to 1 in Hardy-Weinberg equilibrium. What does A and B represent? Consider capital A as the dominant allele, small a as the recessive allele. Alright? Capital A is the dominant allele, small a is the recessive allele. Oh no, because in the equation also we are writing A and B. Let us say capital T and small t. Alright? P plant we are familiar with capital T, small t, 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 T. So, A plus B is equal to 1. What is A representing? Capital T. B is representing small t. So when we are writing A plus B is equal to 1, we are talking about the frequency of the alleles. A plus B is equal to 1 talks about allelic frequency, how frequently the allele is appearing in the population. A square plus 2AB plus B square is equal to 1. A square is capital T, 2AB is capital T, small t, B is small t, small t. This is the frequency of genotype. This is the genotypic frequency. Homozygous dominant individuals are represented by A square. Heterozygous individuals are represented by 2AB. You should not divide it by 2 again. 2AB completely is representing the number of heterozygous individuals, and homozygous recessive individuals are represented by B square. That's all. So this is A plus B is equal to 1 is a representation of allelic frequency. A square plus B, 2AB plus B square is equal to 1 is a representation of genotype frequency. Allelic frequency and genotype frequency. Given below are some stages of human evolution, arrange them in correct sequence. Past to recent. The recent is Homo sapiens, so B should be the end. Correct? Recent is Homo sapiens, B should be the end. Before Homo sapiens it was Neanderthalis. So CB should be the last. CB should be the last two. Oh. Before Neanderthalis it was Homo erectus, and before that it was Homo habilis. What is the name given to each of these? Homo habilis is the tool maker. Homo habilis is the tool maker. Homo erectus is the upright man. Homo neanderthalis is the Java ape man, no? Java ape man is also Homo neanderthalis, Neanderthal man. Homo sapiens is the modern man. What is that which we have got evolutionarily, which is the greatest boon and the bane for human beings? Brain, huh? All other animals have brain, others also have brain. What is the boon and bane for human beings? Boon is because that has given us the ability to create. Bane because most of the human beings don't know how to handle it. It's like you have a very powerful gas lighter, tremendous amount of fire comes out of it. Very often you are keeping it under your seat, burning yourself. That's how it is happening. What is it? Prefrontal cortex. Prefrontal cortex. That is the most advanced evolutionary development in living organisms. Prefrontal cortex is the most advanced neurological development in terms of evolution. That is what makes us logical, analytical, that makes us think, plan, strategize, go back into the past, reflect on the past, project onto the future, and destroy the present. That is what is happening, no? A dog is happy if you just give him what he wants at that moment. He doesn't look backwards. You might have not given food for the dog for 2 days when you had gone on a vacation. And once you come back, the dog is happy. He doesn't remember that. But if it was humans, remember what did you do then? 10 years ago? When I came home, you did not give me a glass of water. You were watching serial. That serial completed 10 years ago, but that serial, your serial is continuing in your mind. One episode, one scene is continuing for 10 years. That's the power of prefrontal cortex. So who is the one who can handle it? The one who can live in the present moment. If you want to harness the power of the prefrontal cortex, you should stop visiting the past or projecting into the future. Just do what you can do to the best of your abilities this moment. That's all. All. Life is simple. Following are the stages of the pathway for conduction of an action potential through the heart. Correct sequence again. Where does it begin first? See, it has to begin somewhere, no? The beginning is SA node. So E should be the beginning. SA node 2, AV node 2, Purkinje fibers, AV bundle. SA node 2, AV bundle and AV bundle 2. SA node to AV bundle to AV node to Purkinje fibers and bundle branches. No, bundle branches and Purkinje fibers. So E, A, C, D, B. E, A, C, D, B. E, C, A, correct, okay. See, interventricular fibers. Interventricular fibers. AV bundle. AV bundle is what? What is atrioventricular bundle? You can consider that as the internodal fibers. AV bundle is the internodal fibers. Atrioventricular bundle is the AV internodal fibers. Okay, so it should be SA node to AV bundle to AV node to bundle branches and Purkinje fibers. But the option is not given there. It should have been EACDB, but we can make it ECADB because E and DB should be in the right places. Anyway, so next, which of the following factors are favorable for the formation of oxyhemoglobin in alveoli? We will do this one and break for dinner. Which of the following factors are favorable for the formation of oxyhemoglobin in the alveoli? High PO2 and lesser hydrogen ion concentration. Now, low PCO2 and high hydrogen ion concentration will lead to low PCO2, it's fine. High H plus concentration will lead to oxyhemoglobin dissociation. Low PCO2, good. But high temperature will lead to oxyhemoglobin dissociation. If oxyhemoglobin dissociation happens, what will happen to the oxyhemoglobin dissociation curve? Increased dissociation, increased demand. See, some points of strategy that we discussed now. Rahul was asking some points of strategy. I'll take 2-3 minutes to reinforce that to all of you. Important. There are some traps that you can fall into at this stage when you see 50 days left. Trap number 1. I am weak in a few chapters. I will do those chapters. That's a trap. Don't fall into that because as you start going chapter by chapter, more chapters will start appearing like you have not completed it. You will fall into the trap of going chapter by chapter. Don't do it. Don't do it. You will not be able to complete all chapters. Okay? You might be tempted to do the other papers also. I have told you do 1 to 12, 51 to 62. Other than that, I can give you another 6 papers if you want to practice it after the complete revision has happened. That you can do. You can take one paper as a final test practice in the month of April also, one paper in 4 days, 5 days. In 5 days you take one paper, so in 30 days you will complete 6 papers. Don't do one in 5 days only. Just to stay in touch with the paper solving also, the flow. To be in touch with the flow, over and above the 62 papers that you would have already solved, I'll give you 6 more papers, 4 or 5 or 6 more papers. So once in 5 days you solve one paper, don't do much, you can do analysis of that also. Once in 5 days when you do, you can do analysis of that also. So we stay in touch with the practice of paper solving also during the month of April. 1 to 12, 51 to 62, if required, whatever additional papers I give. That's all. 13 to 15, if you have already done it, if you have already done analysis of that and made notes from it, great. If you have not done it, don't do it. Alright? If you have not done analysis of the papers from 13 to 50, don't do it anymore. Alright? Stop it. Don't do any more papers for that. All of you have done your perspective notes. Spend 2 or 3 hours every day to revise your perspective notes, linear notes. That will make your concepts more deeper, more stronger. Many questions will be based out of that directly. Something that you have to recognize, something that you have to be familiar with. Your perspective notes will help. 2 to 3 hours every day for revising your multi-perspective notes. 1 to 13, 51 to 62. If I give you the same questions or related questions from these papers, you should be able to get 70-80. That is your approach. You should master everything that is there in 1 to 12 and 51 to 62. Even if you master all the concepts that are associated with paper number 1 to 12. Now I have only discussed 1 to 12 now. I have discussed all the papers. Now we will be completing 12 today. You should be able to get all questions right if I give questions from these papers. If you are not able to do that, stick to mastering paper number 1 to 12. That is highest priority. 51 to 62, priority number 2. Parallel priority is your linear notes, the notes you have prepared, perspective notes. Glossary, extremely important. Glossary of all the 3 subjects, very, very important. If I take that notes from you and ask you questions from your own notes, will you be able to answer everything? Just glossary. Can you? I've been telling this, no? This notes is what you have to repeatedly revise. Even if somebody wakes you up from your sleep, your linear notes that you have made, multi-perspective notes, that should be clearly in your head. If you have not spent enough time mastering that, spend time on that. Okay? From the beginning, I have been telling you strategy only. I have been telling you strategy, what to do at what time. Follow that. If you have followed, very good. You have followed, all of you have followed to an extent. That is reflecting in the improvement that you are seeing in your performance and your confidence also. But all of you have done your own things also. You have done more of what you feel is right than what I have been telling you should be done. Correct? Last 50 days, stick to what I tell you. Alright? This is the best possible approach for you for the last 50 days. If you feel, no, I will do chapter by chapter revision, I will master it, it is not going to happen. It will not work out. As we get closer to the exams, we have to narrow down our range of revision. You cannot have a broad range that you revise from. It is not possible to do that. Alright? Paper number 1 to 12, master everything. Leave the things that you are not confident about right now. Don't do any new learning. 1 to 12, master. That's all. I'm repeating it again and again. 51 to 62, master. Additional papers, give it with your best approach. Correct? Do that. Linear notes, perspective notes, extremely important. 3 hours every day for your own notes, perspective notes, and repeated revision from 1 to 12, 51 to 60. And while attempting the paper, you are at, till January 25th, we were thinking that the question paper will be of 200 questions, 20 options, 200 minutes. But they changed it in January. So you all have practiced for 200 minutes. For the most of the time that you have practiced, it is for 200 minutes. Now you have to cut short into 180 minutes. Again, I tell you, you have to adopt a multi-level approach for solving the questions. One mistake that you can do, like he told, going through the entire paper once to see how the paper is. How will it help you solve the paper? You will still have to go through every question to solve it, no? As soon as you are allowed to open the paper, start from question number 1. Start from question number 1. If you have problem solving numericals, in the first round, don't touch any numericals. You see numbers in the question, leave it. You see numbers in the question, leave it, I am telling you. If you leave it out, then in physics and chemistry also, questions related to theory are like biology only, no? Either you know it or you don't know it. If you are able to apply some logic, you should be able to get answer. Let us say in about 70 to 80 minutes you have completed 100 questions. You are ahead of time. That 20 minutes that you have gained or 10 minutes that you have gained by after completing round 1 will give you confidence. More confidence means more focus. You will get more relaxed. Your mind will relax and focus improves. You will become faster. But if you take it as an ego trip and want to solve every question before you move to the next question, or you are trying to look at the paper to get a feel of how the paper is, from the first question to the last question, you are just browsing through. What if your attention goes to those questions which appear to be difficult? What will it do to your confidence? You will start feeling that the paper is difficult. Even if it is for 2 or 3 minutes that your mind goes down, there is residual effect. During the examination, we don't want any anxiety to come into the picture. Anxiety will make you restless, restlessness will make you hurry, and when you are hurrying things, you are likely to commit mistakes on the easier questions. Because when you hurry, you want to complete the easier questions faster so that you get time to attempt the difficult questions. Don't do that. It is like deciding before the match begins that you will score 350. And you can't. By the way, India won Champions Trophy. Do you know? No. Good. So it's like that. Don't do that. Question number 1, you notice it's a solvable question. Do it. 30-40 seconds, you are done with it. Go to question number 2, numericals, leave it. 3, 4, whatever you can solve. Once you have solved, put a tick mark on the question that you have done it. What is to be done? You can circle it or you can mark it as B, C, whatever you want. Second round or third round. If you circle it, you have to come back again. Whatever has been circled, you come back again. And once you get an answer, let's say question number 1, option D is the right choice. Immediately bubble it in your OMR sheet. Another big mistake many students do is solve the question paper first, mark answers on the paper, and then transfer it at a time. That doesn't give you any significant advantage. It will make you go back and forth to the question paper and the OMR sheet. You will go to the question paper, even the question that you have already solved, you will go back second time to bubble it. Isn't it? So you have solved question number 1, option D is the right choice. You already spent time on that. Option D, you just have to go here and bubble option D in front of the appropriate question. But if you have solved it here and you come back, this time will anyways have to be spent. No, you have to spend time on bubbling. That time you are not saving. But you come back to the question after you have already solved it, and after some more time you come back to the question. You will not just look at it and say question number 1 D, you mark it. Your mind will again want to cross check. That is the nature of the mind. It wants to cross check and see whether it is right or wrong. You are spending double time by doing that. Don't do that. You get an answer for a question, bubble it up immediately. Alright? In these 50 days, what you should avoid is committing mistakes on the questions which you are not supposed to commit mistakes, which on a good day you are getting it right. How will you commit mistakes? Anxiety, restlessness, fear, over expectation. You are going in with an expectation of getting a particular score. You are going in with an expectation that I will first attempt biology, I will complete biology in 60 minutes so that I get more time to solve numericals in physics. Don't do that. 1 to 180. All that you can solve, keep solving, go keep moving. Come back, don't touch the ones that you have already solved. Once you solve it, you know this is the answer, mark it, bubble it, go to the next question, you get the answer, mark it, bubble it. You don't get the answer, circle it, move to the next question. Don't waste time on any question. 1 minute, 2 minutes, 10 seconds, 20 seconds, either ways is important, significant. You lose 2 minutes because you want to get a particular question right. That 2 minutes can destroy your 30 minutes. So now it is strategy. Performance on the day of exam is strategy. You practiced it. And finally, you have to back yourself up. You have to trust your preparation. You have to feel good about the attempt that you give. You should go in with an approach that I'll give my best. I think I am prepared. I'll give my best. That's all. So the remaining papers, whatever you will attempt with that mindset. See, I have a track record of 100% outcome for everybody who follows my advice. 100% outcome. It is not about how much you know. It is about how can you play on the day of the game. Alright? Your target is not 720 now. 720, if it happens, let it happen. You cannot go on to the cricket field telling that I will score a century in 30 balls. If it happens on a good day, great. Why should it not happen? Tell me. If you are able to get 150 questions right by confidence and 30 questions you mark by calculated guesses, on a good day, why shouldn't 30 calculated guesses go right? Why shouldn't it go right? It can go right now. So our approach is to achieve about 70-80% efficiency. Beyond that, there are other factors at play. Alright? Let that play. We will target to get 150 right by confidence. So when you are yourself giving a margin of 30 questions, that gives you relaxation. Alright? You should be ready for minus 180. That's the ideal negative score. Then anything is a bonus. So please be strategic. Don't be in over expectation. Don't try to overdo things. Don't lose sleep. Don't keep studying throughout the night in the room. In the next 50 days, as much as it is important to prepare strategically, revise tactically and go and tackle the exams with confidence, it is important to take care of your food and sleep. Surya Namaskara and Pranayama, what we have done initially, will help at this stage. Spend time on that. Play. Instead of during those 1.5 hours play, instead of playing for 1 hour, you can play for 40 minutes, 30 minutes. But play. Don't disrupt the schedule too much. As it is, you have a 11-hour schedule. Correct? You have a 11-hour learning schedule. You can gain 1 more hour, 1 or 2 more hours during the day. Enough, 12, 13 hours, more than enough. Alright? So, stick to it. Things will work out. Whatever happens, happens. Don't bother about it. Alright? I know when the numbers become 2-digit, in the lower half of those digits, 50 days, or it start becoming 49 days, 48 days, 47 days, anxiety picks up, but you should counsel yourself, tell yourself. It's nothing different. Like Dhoni says, whether it is the first match, or the qualifier match, or a warm-up match, or the World Cup finals, it's after all a match, no? I heard Roger Federer's some university convocation speech, just 1.5 minute speech. He says, you see the efficiency of getting the shot is 60-70%. Roger Federer is considered one of the all-time greats of tennis. His shot efficiency was some 60% points. Every time he plays, about 60% of the times he has got the point. That means about 40% of the times the opponent has got the point. So he cannot keep worrying about, I lost a point. It's just a point. Focus on the next point. You got a point. He cannot be jumping in joy about such a beautiful ace, such a great shot. It's just a point still. Move on. Okay? Okay. So not 62 papers, your mastery will be for 24 papers. That's all. Everything else that you have done, consider it as a bonus. If you have analyzed every paper that you have attempted, excellent. If you have not done it, don't try doing it now. Instead of trying to analyze every paper, master 24 papers. Okay? 168. Matching again. Alpha-1 antitrypsin. Alpha-1 antitrypsin used in the treatment of emphysema. Emphysema is collapse of the bloating of the lungs. Alveoli become bloated, loses their elasticity. Alpha-1 antitrypsin is used for the treatment of emphysema. Cry1A-B, Cry1A-B, Cry1AC, enzyme replacement therapy. Now, 1AB and 1AC is for which one? Cotton bollworm and corn borer. We know it is for one of the two. So let's check whether the other two can give us the right choice. Alpha-1 antitrypsin is for emphysema, A is for 3. There are two options there. D is 2. We got it, no? So even without knowing whether Cry1A is for what, Cry1A is for corn borer, Cry1C is for, 1AC is for cotton bollworm, even without knowing that, you still get the answer. Now you know it. ADA deficiency is what? ADA deficiency. What is ADA? Adenosine deaminase. Adenosine deaminase deficiency leads to the disease called SCID, which is severe combined immunodeficiency. Assertion reasoning. FSH acts upon ovarian follicles in females and Leydig cells in males. True or false? True or false? FSH? False. What does it act on FSH? No? Sertoli cells. FSH stimulates Sertoli cells. It will stimulate the germinal epithelium to multiply. Okay. Reason: growing ovarian follicles secrete estrogen in females while Leydig cells secretes androgen in males. Is it true? That is true. Growing ovarian follicles secrete estrogen, Leydig cells secrete androgens. R is true, A is false. Okay, just tell me the sequence of hormones associated with menstruation acting on the ovary but not produced by the ovary. FSH and LH. The other two hormones of ovary of the menstrual cycle are estrogen and progesterone. Both estrogen and progesterone are produced by the ovary. Estrogen is produced by the growing follicle. Progesterone is produced by corpus luteum, which is the which is the follicle which has released the ovum. Restriction site in E. coli cloning vector PBR 322. Find the role of X and Y genes. PBR 322. X and Y. What is the role of X and Y? X is for over, yeah. Two regions are missed out. One is origin of replication and the other one is copy number. Origin of replication. The gene X is for protein involved in replication of plasmid and Y for resistance to antibiotics. We don't know Y is resistant to antibiotic or not. Gene X is responsible for replication site and Y is responsible for antibiotic resistance. Gene X is responsible for resistance to antibiotic, Y for protein involved in plasmid replication. X for controlling the copy number of the linked DNA and Y for protein involved in plasmid replication. What is correct? Option 4. So copy number and plasmid replication. PBR is what? Plasmid of PBR. Plasmid of some Rodriguez, no? Plasmid of Bolivar and Rodriguez. Okay. Matching again. Cocaine, heroin, morphine, marijuana. Which one can you catch hold of as the right one? Effective sedative in surgery, cannabis sativa, erythroxylum coca, papaver somniferum. It's easy only. Cocaine is erythroxylum coca, A3. 3 options are there. Heroin is heroin. Okay, which one is used as a sedative in surgery? Morphine. Morphine. C1. A3C1. Only one option is there. Option 4. Cocaine is papaver erythroxylum coca. Heroin is papaver somniferum. Morphine is sedative in surgery, and marijuana is cannabis sativa. It's also called hemp plant. Marijuana is called hemp plant. Narcotics are available very freely nowadays. I don't know. It might be a big source of income for the law enforcement also. More crimes, more income for law enforcement. Consider the following statements. Annelids are true celomates. Is it true? Annelids are true celomates. Correct. Who is pseudocelomate? Ashwagandhas are pseudocelomates. Okay. A is true. Poriferans are pseudocelomates. We don't talk about coelom at all. There is no coelom or anything like coelom. There is no body cavity for poriferans. Ashwagandhas are acelomates. Ashwagandhas are pseudocelomates. Platyhelminthes are pseudocelomates. They are acelomates. Only A is correct. 2 statements. In the nephron, descending limb of loop of Henle is impermeable to water and permeable to electrolytes. True or false? Descending loop of Henle. What is it permeable to? It is permeable to water and impermeable to electrolytes. So statement 1 is wrong. Statement 2. The proximal convoluted tubule is lined by simple columnar brush border epithelium and increases the surface area for reabsorption. True or false? False. What is it lined by? Cuboidal epithelium. Cuboidal epithelium with brush border. So both statements are false. Again matching. Joints. Fibrous joints, cartilaginous joints, hinge joint, ball and socket joints. Easy. Catch hold of ball and socket. Humerus and pectoral rotational movement, D2. Only one option. Okay. Fibrous joint, skull, no movement. Cartilaginous joint, cartilaginous joints, adjacent vertebrae, limited movements. They are called intervertebral joints, limited movement. Hinge joint, knee helps in locomotion. Which of the following is not a natural or traditional contraceptive method? Not a natural or traditional contraceptive method. Lactational amenorrhea, is it? It is a natural contraceptive method. Warts are barriers which have to be externally inserted. So warts are not that traditional method. Coitus interruptus and periodic abstinence are all traditional contraceptive methods, not effective but they are used. How do you calculate the date of ovulation? How do you calculate the date of ovulation by the cycle? Today is what date? Today is, what is the date? 13th. Today is 13th. If today is the date of menstruation, beginning of menstrual cycle, that is the first day of bleeding. What was the date of ovulation in the lady in the previous cycle? What was the date of ovulation? What if the cycle was of, what is per cycle is of 40 days duration? Today is the date of, today is the date when the lady has had her first day of flow. The lady has a regular cycle of 40 days. What is the date of ovulation in the concluded cycle, previous cycle? Date of ovulation? 26th, right? 14 days before? 14 days backward from which day? Today. Whatever is the length of the cycle, whatever is the duration of the cycle, ovulation happens 14 days before the commencement of the next cycle. Or after if today is ovulation, 14 days later bleeding will start. Even if it is 20 days cycle, 35 days cycle, 40 days cycle, 28 days cycle, it takes about 14 days after ovulation for the next cycle to begin. So the second half, post-ovulation duration is constant. Variation is in the pre-ovulation duration. Pre-ovulation duration. Question can be, 3 women, A, B, C, have 28, 30 and 40 days cycles. 28, 30 and 40 days cycles. If all of them had their the first day of bleeding on 16th of a particular month, 16th of March, let us say, the date of ovulation for each of the ages again, leptotene, zygote, pachytene, diplotene, diakinesis. Every time meiosis question comes, prophase 1, recall the sequence. Leptotene, okay, what happens in leptotene? Leptotene, bivalent. Zygotine, leptotene is the zygotine is tetrad. Pachytene, crossing over. Diplotene, diasmar. Diakinesis, separation. Now look at it. Diakinesis, completion of terminalization of chiasma, A2, only one option. Cross check with the others. Pachytene, appearance of recombination nodules. Zygotine, synaptonymal complex form. Leptotene, chromosomes look like thin threads. Okay. Again matching. Common cold, hemozoin, viral test, allergy. Which one do you want to catch hold of? White dart test. White dart test is for typhoid. C2. Oh, there are two. Allergy. Dust mites. C2, D4. Both of them are the same. So it is between A and B. Common cold. Rhinovirus. Easy to fix it. Hemozoin. Plasmodium. What color is hemozoin? What color would be hemozoin? Brown color. Malarial parasite eats up the hemoglobin and it produces hemozoin. Assertion reasoning. Assertion. Breastfeeding during the initial period of infant growth is recommended by doctors for bringing up a healthy baby. True or false? True. Why? Because colostrum contains several antibodies absolutely essential to develop resistance for the newborn baby. Reason is the correct reason. A and R are true and R is the explanation. Correct explanation. What type of antibodies are present in colostrum or breast milk? IgA. IgA. What antibodies can cross placenta? IgG. Which antibodies are the biggest pentavalent antibodies? IgM. Which antibodies are produced during presence of which antibodies will tell you the infection is recent? IgM. IgG will tell you that the infection has been a while. It has been a while after the infection. So it is memory-based immunity. If you find IgG in the blood, then it means that your body has already developed a memory for it. So when you test for dengue, you test for both IgG and IgM. IgM will tell you whether you are currently having dengue viral episode going on. IgG will tell you whether you had earlier contacted it, or if this is the first time, have you already developed the memory for it. Again matching. Pterophyllum, Myxine, Pristis, Exocetus. Oh, what do you want to catch hold of? Pterophyllum is angelfish, A3. There are two options. Okay. Myxine, hagfish. Pristis is sawfish. So 3, 1, 2, 4. Option 4. Exocetus is flying fish. The TI plasmid of Agarobacterium tumefaciens stands for tumor inducing plasmid. Tumor inducing plasmid. Why do you induce tumor? What is the advantage of that? It will lead to multiplication of the cells. The TI plasmid will lead to multiplication of the cells, thereby increasing the number of cells with this plasmid. Which of the following is not a component of the fallopian tube? Infundibulum, ampulla, fundus, isthmus. Uterine fundus, isthmus. Infundibulum, ampulla and isthmus are all parts of the fallopian tube. Fundus is not a part of fallopian tube. Fundus is of the uterus. Which part of the uterus is fundus? The lower part or the middle part or the upper part? Upper part. Where else do you find fundus? There are 3 organs in the body which has fundus. Uterus, stomach. The upper part of the stomach is again called fundus. Which one? Which other? There is one more fundus. You don't want to see it because it is out of syllabus. Where else do you find fundus? Ice. That was a clue. You don't want to see it because it is out of syllabus. Was a clue. Okay. Fundus of the retina. They do that, no? Fundal exam, fundoscopy. That small handheld device the device, only the device separates your eye from the ophthalmologist side. Have you not got that test done? Endoscopy? No? Again, matching. Now, genetic disorders. Down syndrome, alpha thalassemia, beta thalassemia, Klinefelter syndrome. Which one do you want to catch hold of? Down syndrome, A1, done, over. Alpha thalassemia? A3, na? Okay, whatever. Down syndrome is 21st chromosome, trisomy 21. Alpha thalassemia is? Alpha thalassemia? Klinefelter is? X chromosome. Alpha thalassemia? 11th chromosome. And beta thalassemia? 16th chromosome. Or is it reverse? Beta is 11th and alpha is 16th. Beta thalassemia is 11th and alpha thalassemia is 16th. Following are the statements about non-chordates. Pharynx is perforated by gill slits. Is it correct? Perforated gill slits. Pharyngeal gill slits. That is the pharyngeal gill slit. That is a reference to pharyngeal gill slits. It is a characteristic of chordates, not non-chordates. Notochord is absent. Is it correct? That is correct. So any state, the combination should have B but not A. Check for that. So 3 and 4 ruled out. Between 1 and 2, let's see. Central nervous system is dorsal. Is it correct? Dorsal central nervous system is a characteristic of chordates. Chordates have dorsal central nervous system behind, pushed backwards. So C is correct. C is wrong. So it is B, D, E. Heart is dorsal if present. Postcranial tail is absent. All those are correct about non-chordates. Again, matching. All chapters they have given matching questions. Mesozoic era, Proterozoic era, Cenozoic era, Paleozoic era. Who developed where? Mesozoic, Proterozoic, Cenozoic and Paleozoic. Mesozoic era, lower invertebrates. Paleozoic era is birds and fishes, birds and reptiles. So D3, only one option. A12, 1, 2, 4, 3. Mesozoic era, lower invertebrates. Proterozoic era, fish and amphibians. Cenozoic era, mammals. Paleozoic era, birds and reptiles. Again, statements. Cerebral hemispheres are connected by a nerve tract known as the corpus callosum. True or false? True. Corpus callosum is grey matter. True or false? Corpus callosum is grey matter. True or false? Grey matter is made up of axons. True or false? Grey matter is made up of cell bodies. Corpus callosum is connection between the two hemispheres. Is it grey matter? It is axons between the two. It is to and fro movement of signals. So grey matter is present in the other part, the cerebral cortex. Connection between the two hemispheres is by corpus callosum, which is axons carrying messages from either side. Statement 1 is correct. The brainstem consists of the medulla oblongata, pons and cerebrum. True or false? Statement 2? False. What is wrong about statement 2? Cerebrum is not a part of brainstem. Midbrain should have been there. Midbrain, pons and medulla. Statement 1 correct, statement 2 wrong. Correct option A, B, C, D with respect to spermatogenesis. Gonadotropin releasing hormone LH acts on GnRH, LH and A is produced. A is FSH. A is FSH. LH acts on Leydig cells. So B should be Leydig cells. FSH Leydig cells. So over option 3. Leydig cells will produce androgen, formation of spermatids. FSH will act on C is Sertoli cells. Factors and D is permeogenesis. Option 3. What is ICSH? What is ICSH? Two options have ICSH here. What is it? Leydig cells are also referred to as interstitial cells. Interstitial cells. What is ICSH? ICSH is? Stimulating hormone. Interstitial cell stimulating hormone. So what is it? LH. LH is also referred to as ICSH in males. LH is in females. The same chemical is called by LH in females and ICSH in males. Same chemical, different locations, different names based on its role. You are a student here. In your home, you are the child. Matching again. RNA polymerase 3, termination of transcription, splicing of exons, Tata box. RNA polymerase 3 produces, which one do you want to catch hold of? Just look at the list 2. SN short nuclear, SN RNPs. Termination of transcription. So, termination of transcription is brought about by Rho factor. Okay. Splicing of exons, SN RNPs. Termination of transcription, Rho factor, RNA polymerase 3, snRNA, short nuclear RNase, tRNA production. Tata box is the promoter. See, the owner of a business is also called promoter of a business. Name determines what you become. Tata, in the name itself, there is promoter. Tata box, promotion of transcription. TATA. Again matching. Exophthalmic goiter, we discussed exophthalmic goiter today only. Exophthalmic goiter, acromegaly, Cushing syndrome, cretinism. Are a lot of match the following questions. Match the following questions are going on and on and on. This is 200 question paper, no? So you need to make it difficult. The last 15 questions. Exophthalmic goiter. Okay, look at the right list 2. Which one do you want to conclude on? Cretinism, hyposecretion of thyroid hormone and stunted growth. D2, only one option. Cross check with the others. Exophthalmic goiter, hypersecretion of thyroid hormone and protruding eyeballs, correct? B4, acromegaly, excessive secretion of growth hormone. Cushing syndrome, excessive secretion of cortisol, moon phase and hyperglycemia. Cretinism, hyposecretion. Why does cortisol lead to hyperglycemia? What is the role of cortisol? Cortisol is also referred to as what? What hormone is it? Stress hormone. What is its role? to increase blood sugar and increase utilization by cells. It will make more blood sugar available, irrespective of what is the blood sugar level. It will contribute to more blood sugar coming into the blood. What is the role that it will do? It will stimulate hunger. You will eat more so that blood sugar can be increased. It will stimulate better absorption of glucose. It will manipulate utilization of glucose to ensure that there is more availability of glucose in the blood. Okay. So overall, hyperglycemia is brought about by cortisol, but hyperglycemia will stimulate the release of another hormone. Which hormone? Hyperglycemia stimulates the release of one more hormone. Which hormone is released when you are in hyperglycemia? Insulin. What will insulin do? Insulin will push it into storage. Insulin pushes glucose into storage, into the fat cells. When glucose level comes down, this fellow is continuously trying to increase glucose. How? Triggering eating. Increase eating, better absorption, better utilization. Cells are triggered to optimize their glucose utilization. Use only how much ever is required so that you can keep using it for longer. But with more glucose, insulin is triggered. It will push into storage. How? Stress leads to obesity. People who do night shifts, night shift jobs. Any of you have anybody in the family or in your known circles who do night shifts? Notice them. Even if they don't eat much, they'll put on weight. Reason is stress. How to deal with stress? Do yoga. Do yoga and manage stress. So people ask, some jokers are fitness experts. Alright? Internet is filled with jokers who call themselves fitness experts. Yoga will not help in weight reduction. You have to do too much of cardio. Brisk walking is better than running for weight loss. I told you one logic earlier. Brisk walking will stimulate utilization of stored fat. Running will stimulate utilization of glycogen because of immediate requirement. Yoga will help you bring down stress. It will tone up the organs. Endocrine secretions will be toned up. So when there is hormonal balance, hormonal imbalances is a major cause of is one of the major causes of obesity. Stress is a major cause of obesity. These two, when you take care of it, it contributes significantly to stemming into accumulation of fat. Alright. Again, match unicellular glandular epithelium. Which one is unicellular glandular epithelium of the four? Can you fix it? Goblet cells of alimentary canal. A3. Compound epithelium, salivary glands, pancreas, goblet cells, moist surface of buccal cavity. Moist surface of buccal cavity. Very tough epithelium. The buccal cavity is made up is very tough epithelium. Multicellular glandular epithelium. Salivary glands, endocrine glandular epithelium, pancreas. Which is the endocrine component of pancreas? Islets of Langerhans. And the exocrine component is? Out of syllabus. Okay. What? Okay. At least tell me the endocrine cells in pancreas. Alpha, beta, delta. Alpha cells produce glucagon. Beta cells produce. Endocrine is not out of syllabus, no? Exocrine is out of syllabus in pancreas. Endocrine is not. Beta cells produce insulin. Delta cells produce somatostatin. Somatostatin is a negative feedback for the growth hormone. Okay. Again, match the following. Structures used for storing food: ring of 6 to 8 blind tubules at the junction of foregut and midgut. Ring of 100 to 150 yellow colored thin filaments at the junction of midgut and hindgut. Structures used for grinding food. All associated with our friend cockroach. What do you want to catch hold of? Huh? What? Storing food. What stores food? Crop. What grinds food? Gizzard. 6 to 8 blind tubes at the junction of foregut and midgut is gastric ceca. What is it equivalent to in your body? What is it equivalent to in your body? It is equivalent to gastric cecae is equivalent to your exocrine glands associated with digestion. So pancreas, liver, that is equivalent to that. 100 to 150 yellow colored thin filaments at the junction of midgut and hindgut, Malpighian tubules, equivalent to kidneys. Correct statement regarding the juxtamedullary nephron. Loop of Henle of the juxtamedullary nephron runs deep into the medulla. That is correct. They have long loops of Henle. Juxtamedullary nephrons outnumber the cortical nephrons. Is it true? Wrong. Cortical nephrons are more than juxtamedullary. Juxtamedullary nephrons are located in the columns of Bertini. Columns of Bertini are the spaces between the renal pyramids. Renal corpuscle of juxtamedullary nephron lies in the outer portion of the renal medulla. Renal corpuscles are present near the junction of the cortex and medulla. Okay, not in the outer medulla. Again match the following. ECG. P wave, QRS complex, T wave, TP gap. What do you want to catch hold of? P wave is for depolarization of atria. So A3, only one option. QRS complex is depolarization of ventricles, T wave is repolarization of ventricles, TP gap, heart muscles are electrically silent. As per the ABO blood grouping system, the blood group of the father is B positive and the mother is A positive and the child is O positive. Their respective genotype can be father is B, mother is A, child is O. Oh. So genotype has to be heterozygous. Both of them should be heterozygous because both of them have contributed one recessive allele, small I, to the child, which has made the child O. If the father is B of B blood group, mother is of A blood group, which blood group is not possible in the child? Options: A, B, O. Data insufficient. Which option do you want to choose? Data insufficient, we don't know. If the father is AB and the mother is O, which blood group is not possible in the child? If the father is AB and the mother is O, which blood group is not possible in the child? Only O? O and AB both are not possible. Alright? So, if in a family of 4, you have all the 4 blood groups, A, B, AB and O, what is the blood group of the parents? Can you conclude? Can the parents be A and B and the children be AB and O? If the parents are A and B, can the children be AB and O? Yes, why not? A and B, If both of them are heterozygous, then one of the child can be O also, no? Both of them have one recessive allele. That is a possibility of combination. So the ignorance of these things have been plotted in some movies, earlier movies. Father is dark, mother is dark. How is the child fair? Is it possible? Is it possible that the parents are both are of dark complexion and the baby is fair complexion? Possible? It is a polygenic inheritance. There are 3 genes which will determine the skin pigmentation. And skin pigmentation is quantitative inheritance. Quantitative. So it is possible that the father has 4 alleles for pigmentation, 2 alleles for no pigmentation. Mother has 4 alleles for pigmentation, 2 alleles for no pigmentation. So both the father and mother are very dark. No, dark. Very dark is 5 pigment and the negroid is 6 pigment. Okay. The child is possible to have 2 no pigmentation alleles from the father, 2 no pigmentation alleles from the mother. Done, no? 1 pigmentation allele from the father, 1 pigmentation allele from the mother. So 4 no pigmentation, 2 pigmentation. So on the Fair and Lovely, what is that? What is, you get that, no? That strip, shade color. The child will be fair, not very fair. Parents are dark, child is fair. Is it unfair? It may not be. So, it serves you well to be educated even if you intend to go into cinema. Okay? True. 198. 2 statements. Gauss's competitive exclusion principle states that 2 closely related species competing for the same resources cannot coexist indefinitely. True or false? Competitive exclusion states that 2 closely related species competing for the same resources cannot coexist. Huh? It has to be different species. Huh?
Same resources.
It is competing for the same resources, no? Statement 1 is correct, no? What is wrong about it? Oh, it is given different resources. Huh? Okay. 198. Statement 2. According to Gauss's principle, during competition, the inferior will be eliminated. This may be true if resources are limiting. Statement 2 is correct. Statement 1 is wrong. Statement 2 is correct. Elimination happens because of the weakness of one of the species.